各位贵宾、师长以及同学们，大家午安，大家好，我是青山大使吴玉荣，非常荣幸能够担任今天的讲座司仪。我们今天的讲座特别邀请到了胡釗逸荣誉教授，莅临本校代表最高荣誉的惠蓀讲座进行演讲。演讲的主题为 "20 Years of Coronavirus Discovery"。这是一个十分难得且珍贵的机会，相信大家都和我一样，非常期待今天的讲座，也预祝大家都可以收获满满。那在讲座开始之前，先向各位介绍今天的与会宾客，请介绍到的宾客起身向大家挥手，也请各位给予热烈的掌声。首先是主讲人胡釗逸荣誉教授，以及本校杨长贤副校长，生命科学院陈学木陈院长，生命科学院黄介成副院长，生医所徐美玲院长，所长抱歉，生医所张家泽执行长，生科系洪惠芝教授，生化所杨俊逸教授，生化所钟三和教授，也感谢所有师生们拨冗参加。那在演讲开始之前，我们先请胡教授上台，来为本次的演讲海报做签名。胡教授请留步。那同时也为了感谢胡教授千里迢迢的来到本校进行演讲，本校特别准备了一面象征最高荣誉的惠蓀琉璃讲座，要致赠给教授。我们有请到副校长来颁发讲座。谢谢副校长，副校长请回座。那胡教授还请留步。另外，生命科学院也准备了精美礼品，有请陈院长上台致赠。谢谢两位请留步。接下来有请生命科学院所有师长们上台，与胡教授一起合照留念。谢谢摄影师。那也为了纪念这感动的一刻，我们邀请摄影师来到台前，为我们大家进行大合照。谢谢摄影师。接下来我们邀请到副校长上台，来为我们的讲座进行引言。让我们以热烈的掌声来欢迎副校长。
好，胡教授，还有我们生科院，还有我们校内校外的朋友们、同仁们，大家午安。那今天非常荣幸有这个机会来介绍我们这个胡教授哈。胡教授是我简单的说他的一些经历啊，然后再说一点他的一些事迹。我们胡教授是1991年香港大学的医学院的毕业，所以他事实上是个医生呐。然后91年到之后呢，到94年到95年之间，他有到 John Hopkins 嘛，是不是？这个去做短时间的这个 study， 所以跟我们的那个张家泽教授有在那个地方有认识，有一个重复。那他们之间的这个友情是非常 非常深的啦，所以这个是才有透过这个机会，胡教授才会来我们中兴大学啊。那他95年之后就到那个香港大学的医学院去工作了，然后97年开始担任助理教授，然后接着几年内就升做副教授到教授，然后一直都在香港大学的这个医学院。到2012年的时候，他就担任香港大学的微生物系的系主任，所以他一直是在这个微生物系里面啊。然后到18年，然后接着到2020年以后，他就是名誉教授，香港大学的，所以他的资历是非常的这个在那个医学的领域是非常完整的了。那他当时在1995年，这个在香港大学这个医学院这微生物系的时候，他是跟着一个是袁国强嘛是这个教授是国际知名的病毒学家了哈，他是师承那个袁教授那里。然后1995年他其实是刚开始，他刚开始不是做我们现在知道的他所谓那个病毒是非常厉害啊。他当时是跟着袁教授，其实是做细菌。他做细菌，当时做1995年做细菌，主要是跟着袁教授做那个人家骨髓病人移植的时候的感染这个探讨。那他就从那里头发觉了说，从那个病人的粪便你可以分离到一些细菌，所以他就引起他的兴趣。所以从95年开始，他其实是从做 bacteria 开始做起的，然后到了2003那个到2003年他才开始做这个我们说冠状病毒嘛，开始做。那所以他在那个做细菌的时候，其实也发生了一些事就是他当时很喜欢，就像我如果讲杨秋忠老师，大家就会清楚一点嘛，就是他到处去找那个有没有新的细菌。结果他后来有在那个香港的那个一些渔获，就是鱼里面，他找到一些新的一些细菌，他就发布了。就发布之后，就没想到会引起一些纷争，因为新闻一发布之后呢，那个鱼就没有人敢买了，所以造成渔民对他一些抗争啊。所以那个时候就从大概是2003年，但是后来他接着做这个冠状病毒之后呢，就开始一直做下去了。那他在这个到接近2015年那个时候，他开始也做一些真菌，所以他事实上他是横跨了 bacteria、virus 跟 fungus， 他三个都有跨。那在香港呢，很多人称他为叫做病毒侦探，detector of virus 病毒侦探。但是他觉得说，你们都没有跟我解释清楚，我其实不是 only detect of virus， 它是 detect of microbiology， 就是微生物，它事实上是这个叫侦探。那他在整个过程中间，整个一直到现在为止，他发现新的病毒超过100多种，所以最近还在世界上排名就是有关冠状病毒的世界科学家的排名前50个，他就是在排名 top 10的，所以他是非常非常这个有成就的。然后在2021年的那个 High-Side Research， 他也是代表香港有获得，所以他的这个研究的这个事迹是非常多的，表现非常优异。2015年他也是得到一个香港重要的一个 award 那 award 这个我很难用中文讲出来有一个 award 是邵逸夫他们颁的那个 award， 那个 award 非常重要，所以他得到那个 award 2015年。所以整体来讲的话，他的表现在对这个病毒上面的研究，尤其 virus 的研究是非常多的。那他做过非常多，他从蝙蝠里面，甚至他跑到中东，在骆驼身上都找到很多很多的病毒。又后来有叫做新的 SARS 嘛，所以都是他在做这个方面的研究。所以在这方面研究已经做到世界级的我们非常有荣幸请他来了。那他其实有个小故事，就是他事实上他从小他其实是自述说他其实念医学院并不是他自己想要的啦，只是说他可能是父母的期待，所以他就念了。那我想很多人大概都是这样的吧。但是念了之后，念了医学院，慢慢有兴趣，就是做研究。他非常喜欢的是做研究，每天来的时候都不知道会发生什么事，他都会预期 unexpectable， 就是你的实验会怎么样，所以 push 他一直想要做研究，所以他才会从一些什么粪便那边去找一些微生物出来嘛。但是可能很多人不知道他其实很喜欢看一些小说了比如说达尔文密码he like to read novels， OK， 像达尔文密码啦，什么地狱啦， 那你不要以为都是这种好像很这种硬性的侦探性，他也喜欢念理性与感性啊，他都会念哈，所以他阅读书这个非常多。那有一天啊，其实我们会预期，他现在已经写了400多篇以上的科学性的 scientific 的 article， 就是科学性的。我们预期有一天他会写 novel， 就写小说。那个小说可能是我们不能知道的，他可能会写什么？因为他现在 keep secret 了，他绝对不能告诉人家。因为哪一天我们突然间发觉在 market 上有一本小说是我们的胡博士写出来的小说，不管是侦探小说还是科幻小说还是爱情小说，这个其实他已经在写了，只是他不告诉人家。这个我是访问了好多人，我才知道说有这件事情啊。所以这一点的话，他如果有机会，他可以告诉我们他到底想写什么。记者问他都不讲，所以可以告诉我们一点 hint， 然后我们就知道说他将来有一天在市面上我们发觉他有小说的时候就知道。但是我预期他可能会写一个，比如说侦探冒险是病毒上面的什么故事了，因为我们现在这个 COVID-19 非常严重嘛。好，那长话短说，就是我们这个非常荣幸啦，好有机会请到我们这个胡教授来这边哦。那也希望哈，我们也预期他可以持续的永远跟我们在一起，好不好？这个是对我们这个学校和生科院的一大助力啦。所以大家今天一定要对他热情啊，让他觉得大家都很热情的希望他在这边哈。好，这不我们就请加入我们掌声，我们欢迎胡教授。
Good afternoon. Thank you very much for inviting me to come. Thank you for the kind introduction. Your team has collected a lot of information for you. So, yes. So in the next 1 hour, what I'd like to do is to share with you our experience on coronavirus discovery. So basically what I will do is to take you through a journey to visit a number of coronaviruses that we have discovered in the last 20 years. So before we start the journey, I'd like to have a few slides just to give you a brief introduction so as to make sure that we all speak in the same language. So everything began because of SARS. Everything began because of the SARS epidemic in 2003. Now, before the SARS epidemic in 2003, the coronavirus field was a very, very quiet field. Nobody was interested in coronavirus research. Very, very, very few people work on coronaviruses. But SARS woke everybody up. So after the SARS epidemic, suddenly many people were interested in coronavirus research. And that has led to a lot of new coronaviruses being discovered, a lot of genomes being sequenced, so as to enable a lot of phylogenetic studies performed, comparative genomic studies performed, and functional studies as well. So, this is how viruses are classified. Now, so as you know, viruses are classified according to the type of genetic materials they carry. So viruses are classified into DNA viruses and RNA viruses, and in turn into double-stranded and single-stranded DNA viruses and double-stranded and single-stranded RNA viruses. So you can see that coronaviruses here. Coronaviruses are positive sense single-stranded RNA viruses. Now, this shows you the situation of coronaviruses before the SARS epidemic. So right before the SARS epidemic, coronaviruses was like this. Now, this is an electron micrograph of coronaviruses, and this is a schematic representation of a coronavirus. So you can see that on the surface of coronavirus, there are a few proteins, including the SEMN, spike, envelope, and membrane. And then within the coronavirus, this is the single-stranded RNA, all right, because coronavirus is a single-stranded RNA virus. And there's a protein associated with it called the N protein, nucleocapsid protein. Now, this is the genome structure and the classification of coronaviruses before the SARS epidemic. Now, the genome size of coronavirus is around 30 kilobases, all right? So the 30 kilobase, the 5' 2/3 of the 30 kilobase encodes an open reading frame called ORF1AB. Now, this open reading frame is in turn cleaved by its own protease into 15 to 16 non-structural proteins, 15 to 16 NSPs. These NSPs contain some of the very famous proteins like RNA-dependent RNA polymerase, or PO, helicase, the proteases, which are targets of some of the antivirals, and so on and so forth. And then downstream to ORF1AB is the spike gene, which encodes the spike proteins on the surface of the coronavirus. So in the electron micrograph, you always see this spike protein sticking out, giving rise to what we call the crowned appearance of the coronavirus, because corona means crown. And then downstream to the S is the envelope protein, envelope gene here, membrane here, and then the N, nucleocapsid gene, encoding this nucleocapsid protein here. Now, coronaviruses were classified into 3 genera before the SARS epidemic: alpha, beta, and gamma. Actually, they were not called alpha, beta, and gamma before the SARS epidemic, but let's call them alpha, beta, and gamma. Now, the first coronavirus discovered was this one, infectious bronchitis virus, which is from poultry, from birds. This is actually the only bird coronavirus up to the moment of the SARS epidemic. That was discovered in the 1930s. And then in the 1940s, people discovered this one, murine hepatitis virus from mouse. And then in the 1960s, people discovered these 2 human coronaviruses, human coronavirus 229E and human coronavirus OC43. And these 2 human coronaviruses were actually the only human coronaviruses known before the SARS epidemic. So coronaviruses were classified into alpha, beta, and gamma coronaviruses. Alpha and beta are from mammals, all right? So all these are from mammals. Gamma is from birds. So that was the situation before the SARS epidemic. So with this background, let's start our journey to visit a few coronaviruses that we discovered in the last 20 years. So the first one is a human coronavirus HKU1. Now, so we have to go back to 2004. That was after the SARS, right after the SARS epidemic. So at that time, we saw a patient, a 71-year-old male patient who presented because of fever, cough, and sputum. Chest X-ray of the patient showed left lower zone infiltrate. So this patient clinically had a pneumonia, community-acquired pneumonia. So what we did was we performed a standard investigation for a patient with community-acquired pneumonia. So we collected the nasopharyngeal aspirate from a patient and performed various tests to investigate the standard viruses, standard respiratory viruses like influenza, metapneumo, and so on and so forth. All the viruses, but those are all negative. We collected sputum and tried to investigate for bacterial and mycobacterial causes, and were negative. Pair serum to investigate for mycoplasma, chlamydia, antibody, but they were all negative. The patient was discharged on day 5. So the patient became better, discharged, but we couldn't find the cause for the patient's pneumonia. So at that time, because we have been working on SARS for a while, so we wonder whether there is a novel pathogen in this patient, and because we have been working on SARS, we wondered whether this novel pathogen is a novel coronavirus. So what we did was we used the patient's nasopharyngeal aspirate And we extracted RNA from the nasopharyngeal aspirate, and we performed an RT-PCR on the extracted RNA, which is specific for the PO gene for coronaviruses. So we used the pair of conserved primer for coronaviruses. We tried to amplify a 440 base pair fragment. Results were positive. There was a band. We sequenced it, and we found that it is here after phylogenetic tree analysis. So we found that this thing is clustered with the beta coronavirus group. So at that time, because it was after SARS, so there's SARS there. So SARS is actually a quite distinct branch from the other few genera of coronavirus. So SARS coronavirus is another group under beta coronavirus. But we found this one. So we are quite excited because we think this is probably a novel coronavirus. So at that time, actually, the Dutch has also discovered another human coronavirus. They call it human coronavirus NL63. NL stands for the Netherlands because it's from the Dutch. That is an alpha coronavirus, distinct from ours. So what we did was we sequenced the genome of this human coronavirus, which we subsequently called human coronavirus HKU1, and compare the genome with the other known coronaviruses. So we found that this genome structure is actually similar to beta coronavirus lineage A. Lineage B is SARS, all right? Lineage A is the standard beta coronaviruses. So it is similar to beta lineage A with two papain-like proteases with the hemagglutinin esterase, and so on and so forth. Now, so after we reported this new coronavirus, other people in the world also tried to see whether this coronavirus is present in their patients. And the result was yes. It's basically found all over the world. So it's a global coronavirus. It mainly causes diseases in winter. It is a human coronavirus because it's only found in human. There's no annual reservoir. Transmission was from human to human. The incidence is around 1 to 5%. So you can see that subsequently we perform epidemiology studies on these various human coronaviruses. OC43, 229E, which were discovered in the 1960s, remember? This one, this new one, and the Dutch human coronavirus, NL63. So you can see that in our study, like OC43 is the most prevalent. But in the America study, HKU1 is most prevalent. Italian study, 229E is most prevalent, and so on and so forth. So you can see that actually different human coronaviruses are more prevalent in different countries at different time. So this, after we have discovered this human coronavirus actually became a common cause for respiratory tract infection. So you know nowadays there are lots of these kits for multiplex PCR and so on and so forth, which can amplify various respiratory pathogens at the same time. So for this particular kit called FIMARRAY, these are the various respiratory pathogens it can amplify at the same time. So you can see that there are these 4 coronaviruses with HKU1 here, influenza, parainfluenza, and so on and so forth. So that is our human coronavirus HKU1. So now let's move on to the animal coronaviruses. Now, when SARS was first discovered, people also discovered SARS coronavirus in this animal, civet cat. But after a short while, people found that actually civet cats were not the ultimate reservoir for SARS coronavirus, but probably just an intermediate or amplification host for SARS coronavirus. Because of that, we tried to look for the ultimate reservoir of SARS coronavirus, and therefore we performed this epidemiology study in Hong Kong. So in 2004, 2005, we collected more than 400 nasopharyngeal and anal swabs from these various kinds of animals, bats, rodents, monkeys, from many different locations in Hong Kong. So we screened all these specimens using the same pair of conserved primers that we used to screen the human specimen. And we found that some specimens were positive, and we will go to that in a moment. So this is a picture taken from one of our trips to the country park to take bat samples. As you know, bats live in these water tunnel caves. So we have to go into these water tunnels and caves in order to find the bats and take the sample. So this is a bat expert in Hong Kong, and this is the equipment that he used to catch the bats and take the bat samples. So there's a long pole with a net. So what we did was, you know, bats like to hang themselves in the roofs, roosting in the water tunnels and caves. So what we did was we used that long pole to tip the bat so that the bat will fall into the net. So by that, we can catch the bat, and we can use the bat and perform nasal swab, anal swab, and take the sample. This is another piece of equipment that we use to catch the bats, called the mist net. Actually, there is a net here, but you can't see it. The bats can't detect it. So when the bat fly through the net, it will be caught. And that is the reason why it's called the mist net, because it's like the mist. So when the bat is caught, we measure the bat's arm span and take note of the morphology of the bat in order to identify the bat species. But in addition to that, we took nasal and anal samples from the bats. So we used the swab to take nasal and anal samples from the bat, put it in virus transport medium, take it back to our laboratory, and we used the same pair of primers that we use for detecting coronaviruses to try to look for coronavirus in these bat samples. So this is the result. So what we found were 3 new coronaviruses. And at that time, because we saw this, we were very excited because this one looks very much the same as the SARS coronavirus in human and civet. So we thought that we might have found the reservoir of SARS coronavirus. So we disregard this one, disregard that one. We only work on that one. And we sequenced its genome, of course. And we also try to see how prevalent it is in these animal samples. So what we found was this particular coronavirus is only present in one bat species that we found, called Rhinolophus sinicus, Chinese horseshoe bats. It's not present in the other bat species that we collected, not in rodents, not in monkeys, but only in Rhinolophus sinicus, or Chinese horseshoe bats. It is named as horseshoe bat because of the shape of the nose. So you can see that it's like horseshoe, and that is the reason why it's called horseshoe bats. It likes to gather together, roost together inside water tunnels and caves, and that actually encourages a lot of inter-animal virus transmission. So we sequenced the whole genome, we performed phylogenetic analysis, and we found that for the PO gene, it looks very, very much like the SARS coronavirus found in humans and civets. For the S gene, it also looked very much like the human and SARS coronavirus. So that is bat SARS coronavirus HKU3, the ancestor for SARS coronavirus. So now let's move on to some other bad coronaviruses. Now you remember this phylogenetic tree. So in addition to this bad SARS coronavirus, there are actually other new coronaviruses. And our hypothesis was that at that time was probably there are actually more coronaviruses in bats than these two coronaviruses. So because of that, we performed a bigger surveillance study in Hong Kong. We collected more than 600 nasopharyngeal and anal swabs from more than 300 bats of 13 species in 20 different locations in Hong Kong. We extracted RNA. We used the same pair of primers, conserved primers for coronavirus for detecting coronavirus in these bat species. And in this study, we found 6 novel coronaviruses in different bat species, greater bat-winged bat, lesser bat-winged bat, and so on and so forth. And we named these 6 novel coronaviruses as bat coronavirus HKU2, bat coronavirus HKU4, HKU5, HKU6, HKU7, and HKU8. Now, what is most interesting is the phylogenetic analysis. Again, we sequence the genomes and try to analyze their phylogeny. So these in blue and in red are the 6 new coronaviruses. This is SARS. So at that time, we have alpha, we have beta lineage A, beta lineage B, and gamma. So you see that there are some novel coronaviruses which belong to the alpha coronavirus genus. What is most interesting is this group, which consists of 2 novel coronavirus, bat coronavirus HKU4 and bat coronavirus HKU5. You can see that this group is distinct from the other beta coronavirus. It is kind of linked to the other beta coronaviruses, including the classical lineage A SARS coronavirus, but it forms a distinct branch among these beta coronaviruses, and we name it as beta coronavirus lineage C. Now, at that time, when we discovered this new lineage of coronavirus, we didn't know that it is important. We don't know how important it is until MERS emerged in 2012. So when MERS coronavirus emerged in the Middle East, people immediately sequenced the genome of MERS coronavirus. They found in camels. And when they built a phylogenetic tree, they found that actually MERS coronavirus is closely linked with HKU4 and HKU5, the 2 bad coronaviruses that we discovered 6 to 7 years earlier. So these discoveries has actually enabled or accelerated a lot of research in MERS. Subsequently, we also showed that the S protein of this bad coronavirus HKU4 binds to human DPP4, which is the receptor for MERS coronavirus. And recently, last year, we were able to isolate bad coronavirus HKU4 and show that it can actually infect human DPP4 transgenic mice, causing pathology in these transgenic mice. So it shows that this HKU4 is actually really closely linked to MERS, which is able to cause similar pathology as MERS in these transgenic mouse. Now, so after this Hong Kong study, we believe that, of course, there are other bad coronaviruses elsewhere. Therefore, in the next study, we move to Guangzhou to try to perform a surveillance study in Guangzhou. Now, in this surveillance study in Guangzhou, we again collected more bat species of different kinds of bat species. Again, anal swab mainly and some nasopharyngeal swab. We use the same pair of primers. So of course, in this surveillance study, we can detect some coronaviruses that we have already found in Hong Kong, like HKU2, bat SARS coronavirus HKU8. But in addition to that, we also found 2 new coronaviruses. We call them bat coronavirus HKU9 and bat coronavirus HKU10. Now, of course, what is most interesting is, again, the phylogenetic analysis. So alpha, beta, lineage A, B, and C, so HKU4 and HKU5. Lineage B is SARS, gamma. So you can see that here, there's another group which consists of a branch which is distinct from the other beta coronaviruses, which are the bat coronavirus HKU9. So this particular group is actually a new lineage under the beta coronavirus. So we called it beta coronavirus lineage D. Now, before we studied coronaviruses in bats, actually we didn't know that much about bats. But because we have found some new coronaviruses in bats, we tried to start reading up a little bit about bats, and we found that actually bats are not that far away from us. We use bats for various purposes. So what you can see here are the fecal materials of bats, and they are actually used as Chinese medicine. So as you can see here, like for this one called Ye Ming Sha, so it implies that this particular medication was given to patients who had night blindness. So that is the name Ye Ming Sha. All right. So that means if you have night blindness, and if you go to a Chinese medical practitioner, he may prescribe you with these bad feces, and you need to take it in order to cure your night blindness. Now, bats can be used as food. This is a picture that we took when we went back to Guangzhou. When we went back to Guangzhou, I deliberately asked them to bring us to these, you know, in Guangzhou there are lots of wet markets, to bring us to one of these Chinese wet markets. All right, 野味市场. I deliberately ordered this dish. This is bat, all right? Of course, I did not eat it. I ordered it just for the sake of taking a photo. So, but bats can be used as food, all right? And it is present in these Guangzhou wet market restaurants. This is bat minced meat, 肉饼, all right? And we have hot pot. I know you guys love hot pot. Taiwanese love hot pot. So basically they put the whole bat in the hot pot and for 火锅, all right? Interesting. And because of these Chinese wet market, these are the places where there are many live animals. And a lot of mixing of these live animals and also mixing between humans and these live animals. And it encourages a lot of interspecies jumping. As you know, the first victim of SARS coronavirus in 2002, 2003 was actually a cook in Shenzhen. And that may be the reason why it has jumped from one species to another, causing the SARS epidemic in 2003. Now, so that's bat coronaviruses. Let's move on to another group, bird coronaviruses. Now, you remember, in one of the very first phylogenetic tree that I showed you, you remember the first coronavirus discovered was called infectious bronchitis virus. It was discovered in the 1930s. It was the only bird coronavirus before the SARS epidemic. But because we have found so many bad coronaviruses, so at this point, we thought that there might be actually more bird coronaviruses in other bird species, because you know there are a lot of bird species. So because of that, we carried out a surveillance study in Hong Kong. So we collected more than 1,500 dead wild birds from 77 different species in 32 families in Hong Kong. Again, we collected nasal swabs, cloacal swabs from these birds, used the same pair of primers that we used for all the previous studies, and tried to look for novel coronaviruses in birds. And what we found here were 3 novel coronaviruses. We call them Bubu coronavirus HKU11, thrush coronavirus HKU12, Munia coronavirus HKU13. So these were the birds that from which we found these novel coronaviruses. Again, what is most interesting is the phylogenetic analysis. So alpha, beta, lineage A, B, C, D, gamma. So for gamma coronavirus, actually at that time there is a new coronavirus called beluga whale coronavirus. This is very interesting because it's from mammal. Previously, this group of coronavirus was only found in birds, but people in Europe found it in a marine mammal. They called it beluga whale coronavirus. But for our HKU viruses, this form a distinct branch among all the coronaviruses. And actually, this represents a new genus coronavirus. Before that, before the SARS epidemic, we only have alpha, beta, gamma. We now have a new genus of Delta coronavirus. Now, with so many coronaviruses, so many genomes, in addition to phylogenetic analysis, we can also perform comparative genomic analysis. So this just briefly tells you that you can see that some coronaviruses has 2 papain-like proteases, some have only 1, some have the hemagglutinin estrates, and so on and so forth. And we can also compare the genome size and so on. Now, because actually there are many bird species, but we only found 3 novel bird coronaviruses. But we found so many bad coronaviruses, so we were not that happy. So we tried to perform another surveillance study. This time, we didn't use the conserved primers for coronaviruses in general, but we designed a new pair of primers which are conserved just for the Delta coronaviruses, because it's not that conserved. So theoretically, we can pick up more, because if it is more conserved, it's more degenerate, it's less sensitive. So because we don't add in that many degenerative bases, so it becomes more sensitive, and we may be able to pick up more Delta coronaviruses. So in this study, we not only try to detect it in birds, but we also collect mammal specimens, because in gamma they found it in mammal. So in Delta it may also be present in mammal. So we collected both bird and mammal samples. And what we found was a number of new bird coronaviruses in different bird species, and we call them white-eye coronavirus HKU16, sparrow coronavirus HKU17, magpie robin HKU18, night heron HKU19, Wigeon HKU20, and common oran HKU21. But in addition to bird coronavirus, we also found this one in pigs. So we found a Delta coronavirus in mammals in addition to birds. At that time, we didn't know how significant it was, but subsequently people again found it in other parts of the world, and it found that this particular porcine coronavirus HKU15 was associated with a lot of illness in pigs, a lot of fatalities in pigs. So phylogenetic tree construction, of course, they are clustered together, all right? And you can see that this porcine coronavirus is actually pretty close to the sparrow coronavirus HKU17, meaning that it might have jumped from birds to mammal some time ago. And more than that, we also found this coronavirus HKU15 not only in fecal samples of pigs, but also in the respiratory tract. So this is another new finding. Now, after this, after we have discovered these bat and bird coronaviruses, we tried to build an evolution model for coronaviruses. This is our evolution model. So what belief was coronaviruses somehow, some time ago, infected either a bat or birds. And then sometime down the line, it jumps from bats to birds or birds to bats. But in the bat lineage, it of course infected other bats. And it also infected mammals, humans, and so on and so forth, giving rise to these alpha and beta genera. For the bird lineages, of course, it infected many bird species, but somehow, sometime it jumps from birds to marine mammals. You remember there is a whale for gamma, and it can jump from bats to pigs, and so on and so forth, giving rise to these alpha, beta, gamma, and delta coronaviruses that we see at this particular moment. Now you remember this phylogenetic tree. There is a marine mammal coronavirus here. Of course, our next question is, are these gamma coronaviruses in other marine mammals? So is there a lineage of marine mammal coronavirus? So because we are in Hong Kong, we went to the ocean park to look for collaborations, because in ocean park, there are different kinds of marine mammals. So what we did was we asked them to help us collect different samples from different marine mammals, sea lions, harbor seals, dolphins. And we found a novel coronavirus in dolphins. And we used, the same pair of primers we used for other discovery of other coronaviruses. And we named this coronavirus as bottlenose dolphin coronavirus HKU12. So it is here, all right, gamma coronavirus. There's an avian group and a marine mammal group. This is the beluga whale coronavirus where the Europeans discovered, and we found this dolphin coronavirus in Hong Kong. So comparative genomic studies, so it's here. So you can see that it actually looks very much like the beluga whale coronavirus with one papain-like protease with a very large genome. In fact, its genome is actually largest. The genome of this coronavirus and the beluga whale coronavirus is actually largest, with a lot of potential open reading frames, which may encode additional non-structural proteins. Because this is from Ocean Park, and they collected serial samples from these animals, serial archive samples from these animals. So we can perform serological studies and serial RT-PCR studies. And actually, we can somehow find seroconversion in these dolphins. So we have dolphins here. Now, you know, when MERS emerged in the Middle East in 2012, people also found MERS coronaviruses in dromedaries, in dromedary camel. And that makes me very interested in this particular kind of animal. And the next question is whether there's another coronavirus here, whether there are other viruses, other novel viruses in this particular species. We won't talk about the other novel camel viruses, but we will just take a look at the camel coronavirus. So because it's in the Middle East, so I look for collaborators in Dubai. So I actually flew from Hong Kong to Dubai. Right after Chinese New Year, I went to visit my collaborator. This is the lab. This is the tallest building in the world in Dubai. So this is the lobby of the lab. So we have to, so of course, this is Dubai. So you will have these sheikhs as the leader of the lab. And you can see in the lobby, they are showing various kinds of animals on the television, camel raising, and so on and so forth. All right, camel raising. So you know, in Dubai, there are two kinds of animals which they take as very precious. One is mammal, one is camel, the other is falcon. So we have a story for camel and another story for falcon. So this is the veterinary surgeons that I collaborated with. He is actually the head of this veterinary laboratory. He's actually a German. He went to Dubai around 30 years ago. He stayed in Dubai with his wife. 30 years ago, he told me when he first went to Dubai, his laboratory has only 3 people. He, his wife, and another technician. But now his lab has more than 100 people. So I visited his lab. So it is an autopsy room where they dissect dromedary, camels, as well as other animals. But what I'm most interested is just these fecal samples from these mammals. So I collected these fecal samples, again put it in virus transport medium, and carried them back to Hong Kong. And used the same pair of primers, and we discovered the new coronaviruses in dromedary camels, which is here. So it is basically a lineage A beta coronavirus, which we name as dromedary camel coronavirus UAE HKU23. UAE stands for United Arab Emirates, all right? We performed seroprevalence studies, and we found that actually at least half of the camels have been infected with this coronavirus. So we have a camel here. Now, falcons. So how about Delta coronaviruses in the Middle East? We asked our collaborator to collect samples from various bird species in Dubai, including falcons, many, many bird species. We used the Delta coronavirus specific primers, all right, because it's more sensitive for Delta coronavirus. We particularly try to look for Delta coronaviruses in birds and mammals, perhaps. But in this particular study, we didn't find any coronavirus in mammals, but we found 4 new coronaviruses in the bird species. We named them as falcon coronavirus UAE HKU27, Hobura HKU28, Pigeon 29, quail 30. And you can see that these are closely linked to other Delta coronaviruses. And again, we can perform serological studies, seroprevalence studies, because they have, again, collected and archived a lot of falcon serums, because falcons are really precious in Dubai. And therefore, they took their falcons to see a lot of doctors, and therefore they can archive a lot of falcon serum samples. And you can see that up to 75% of the falcon serum samples were antibody positive. So they have a lot of these meetings where they share experiences with falcon recreation. They used falcons for hunting in the past, but no longer nowadays. They just use falcons for recreations. But all these rich sheikhs, they all have some falcons as pets, and camels as well. So this is the last one. It won't take long. Now, evolutionary origin of lineage A beta coronavirus. Now you remember the evolutionary model. For alpha and beta, it came from bats. For gamma and delta, they came from birds. But very, very interestingly, for the beta coronavirus, in this particular lineage, there was no single bat coronavirus. Actually, up to this moment, no single bat coronavirus were found in this particular lineage of beta coronavirus. And you know, bats are actually the second most diverse mammal on Earth. The most diverse group of mammal on Earth is rodents. So we wonder whether actually rodents may be the reservoir of this particular lineage of beta coronavirus. So that is the reason why we perform a surveillance study in rodents. We collected some different species of rodents and used the same pair of primers, and we found one novel coronavirus. And we called it Chinese rectus coronavirus HKU24. So you can see that it's here. So it may or may not support the hypothesis, but we still have to find more coronaviruses in rodents in order to see whether this is true or not. But this has modified our evolutionary model a little bit. So for the lineage A, maybe it comes from rodents. So there's still work to be done along this line. So that's actually all I would like to tell you about coronavirus discovery in the last 20 years. So I'd like to thank my collaborators. So my co-investigator, Susanna and KY, our clinicians and microbiologists in Hong Kong, because they have helped us a lot in collecting human samples from patients, in particularly in our human coronavirus HAU1 studies. A few departments in our Hong Kong government, because they have a lot of experts on various kinds of animals. And actually they helped us to collect all the mammalian species and birds from the various kinds of mammals, pigs, bats, and so on and so forth from Hong Kong. My collaborator is Ocean Park from Guangzhou, CDC. My collaborator in Dubai and his team. Our laboratory in Queen Mary Hospitals, and of course our graduate students, because they are the people who has really done the hard work. So thank you very much.
Okay, we are really appreciate your presentation. It's really very informative. I'm pretty sure there are lots of questions were raised by this audience. Okay, 你们各位有问题的话，现在我们可以，因为这个时间我们还有很多，我们可以好好的来问我们的这个胡教授。OK， 任何你周三和周老师。
Yes, thank you very much for your talk. I think in your presentation you have described the discovery of many different coronavirus in different wild animals. So my question is, what's the functions of this coronavirus in these wild animals? Why are they present in such wild animals? Do they have any function? Do you know what I mean?
No, coronaviruses, like other microorganisms, most are not pathogenic, but some are pathogenic, like the HKU15, the porcine coronavirus HKU15, that is pathogenic. But for most microorganisms, actually, you know, inside our body we have many bacteria, many viruses which are not pathogenic. So coronaviruses are just like other viruses and bacteria. So in those animals that we have found, basically we have not seen any illnesses in the animals when our collaborators helped us to collect the samples. But of course, those animals might have just been suffered from very, very mild illness that they don't know. Because, you know, for human coronaviruses, actually for 229E, HJU1, and L63, they infected humans, but they only cause very, very mild respiratory tract illness. So for other coronaviruses in other animals, it is possible that they only cause very mild illness, but we are pretty sure that they have infected those animals, because in a lot of animals we can find that they are seropositive, meaning that they have reacted to the presence of the coronavirus. But those are just acute infections. After seroconversions, the coronavirus just go. Like in the dolphins, you can see that the RT-PCR was negative and then become positive, negative, negative. But then there's seroconversion. So it means that the coronavirus did infect the dolphin, causing seroconversion. But it was not a fatal illness. Might be some mild illness in the animals.
Professor Hu, very thanks for your talk. I have a question. Why this coronavirus has species specific? Yeah, it's my first question. And so why, for example, why human cannot be infected by birds coronavirus? Can you explain from this evolutional view? Yeah, thank you.
When coronaviruses infect a particular kind of animals, the first thing that happened is the binding of the S protein of the coronavirus to the receptor in the animal cell surface. So that has some kind of very specific interaction. And most of this species specificity probably resulted from these sort of S protein receptor interaction. So, and that is also the reason why the coronaviruses do not infect human, because humans just do not use that particular receptor in the animals. And even if human has that kind of receptor, the configuration is not correct. So it doesn't cross the species. And actually, this is also true for other viruses. They also have some kind of species specificity. And when they need to cross species, one of the very first steps is surface protein and receptor interaction. They have to overcome this as the first step. But of course, even if they get into the cells, there are many other subsequent obstacles. But the surface protein and receptor is the first step.
Yeah, right. Since there are so many coronaviruses, in fact, many different animals, why there are only 7 coronaviruses ever successfully infect human beings? So there are 3, SARS-CoV-1 and SARS-CoV-2 and MERS. They create serious problem. But why not the other 4 create serious problem to human beings? So what's the difference between these 4 so-called common cold coronavirus and then compared with those serious viruses? And then another question, if I can ask, is for SARS-CoV-1 and SARS-CoV-2, it took probably 1 month or 2 months or several months or several weeks for humans to identify they are coronavirus. However, it was too late because the virus had been spread all over the world and create serious problem. Is that new measure to detect the serious problem at a very quick speed, and therefore we can prevent the spreading of new virus ever emerge and come to human being in the later time? Is that possible? Okay.
So the first question for the human, yes,
yeah,
for the human coronaviruses, why don't they cause serious illnesses? Now, this is just a theory. Usually, during interspecies jumping, when a particular virus first jumps from one species to another, it didn't adapt well. Yes, so right. It is more likely to cause serious illness at that time when it first jumped. But then after adaptation, it became less and less severe. That is the usual story.
Yeah, right.
So you can see in SARS coronavirus 2, at first the mortality was high, but you know when we now have Omicron, the mortality seems to reduce. It's more infective, but it doesn't kill that much.
Yeah, right.
And maybe we have a new SARS coronavirus 2, a new variant. It may be even more effective, but it kills even less. That is the usual story. So for the four human coronaviruses, they might have jumped from animals from animal to human hundreds of years ago. So hundreds of years ago, it may have caused serious illnesses at that time. But then after adaptation in human, it became human coronaviruses. So for the SARS disappeared, and then MERS and SARS coronavirus 2. So after some adaptation, maybe after a few hundred years, it became human coronaviruses then. So the next question is, of course, a $1 million question. Very, very difficult question. To predict an epidemic before it happens is super difficult. Many people try to do that because they try to do many surveillance studies in animals and humans, particularly in humans, because but the story is usually like that. In human, when there is a group of patients with which suffer from an infection with an unknown cause, then people started to look for what the particular cause is. And they found that it is something new. Now, of course, if you want to know the pathogen before it became severe, the answer is a lot of surveillance studies. A lot of surveillance studies. So before the pathogen actually explodes as a pathogen, you might have already detected it in the form of a non-pathogen. So just like the MERS epidemic, actually, as I've told you, we found the bad coronavirus HAU4 and HAU5 6-7 years before the MERS coronavirus epidemic emerged in the Middle East. Actually, that has already helped solve a lot of problems when MERS emerged, because people immediately know a lot about that virus. And that has helped a lot in the basic science research in that coronavirus. But of course, the answer to your question is difficult, because we simply don't have enough money and manpower to perform so much surveillance studies. You know, when SARS emerged in 2003, a lot of money were poured into SARS coronavirus research. But SARS coronavirus disappeared after a few months. Immediately, all the money disappeared. Yeah. So no government is interested in coronavirus research again anymore. They went back to influenza because we have a lot of influenza. So when MERS emerged, then the people have some interest in coronavirus. But somehow they found that it only occurred in the Middle East. You don't have a single case in Taiwan. So your government will not support a lot of MERS coronavirus research. And you know, fighting for those money is a big problem. And not that many people want to spend money on on a lot of surveillance studies.
Thank you.
Thank you very much for your excellent presentation. So one question, as we know, SARS-CoV and the MERS belong to beta, from the beta group from the phylogenetic studies. But the two viruses has a specific interaction with different receptors. For example, like COVID has a interaction with ACE2, but MERS has a specific interaction with DPP4. Does it mean even the different, even the virus in the same phylogenetic branch can interact with different receptor protein in the animal cells? And the second question is, you mentioned a lot about how you take the sample, and you also mentioned you take the fecal samples from the animal. Does that mean that the COVID-19 could be transmitted by the feces?
So let me answer your second question first. Can COVID-19 be transmitted? People have found SARS coronavirus in fecal samples. So theoretically, yes, but definitely it is not a main route of transmission. You know, the respiratory route is much more important route of transmission than the fecal route. So theoretically, wherever you can find the coronavirus, it is theoretically a possible source of transmission. But, you know, for a coronavirus like SARS coronavirus 2, it's everywhere. So you just don't have to worry about it. You basically don't need to wear a mask, to me, because you will get infected today or tomorrow or day after tomorrow. So it is really highly prevalent virus. So so back to your first question, yes. Even though they are phylogenetically, they are pretty near to each other, they can use different receptors, radically different receptors. But actually, for the two examples that you gave, the SARS coronavirus and MERS coronavirus, they are both beta coronaviruses, but they are two lineages. So they are close together in the sense that they are both beta, but they're actually not that close because the S protein sequences are quite different from each other. So yes, they do use different receptors. Even they appears to be a little bit close to each other. Of course, a very important next study to do is to find the receptors of these coronaviruses, but you know, and see the relationship between coronaviruses and the receptors. But as you know, finding receptors is very difficult. So it takes quite a lot of time.
Okay, professors, thank you for your excellent talk. I'm just curious about, like, from the evolution point of view, you mentioned that coronavirus or SARS like tend to become less pathogenic, but has like strong power for infection, right? It could in fact like be more widely infect to people or to human, to like animals. Is this the feature for all viruses, or this is just a common feature for, let's say, single strand positive sense DNA virus?
It tends to be a feature for most viruses because the evolution takes place because of selection. Now, there's evolution because the coronaviruses continue mutate. Because it's an RNA virus, it mutates very rapidly. So after each generation, if the coronavirus infects me and then I pass this to you, there are a few base changes in the coronaviruses. So after some passages, there will be many mutations in the coronaviruses. So theoretically, there are new variants. But which variant will survive? The question is which variant will be able to survive? The variants that survive, of course, will be the variants that is more infective. Because if it's more infectious, it will be present everywhere. So if it's not infective, how can it survive? So why less fatal? All right. The next question is why less fatal? It is because if it causes a lot of fatalities, a lot of serious illnesses, the patients will quickly be isolated. and the virus will not be able to infect further people. And if the patient dies immediately, it is not that easy for that patient to spread the virus to other patients. But if the infection is not severe, the patients can move around. If I am infected with a with a COVID-19 variant, which is very mild, I don't know that I am ill. I don't know that I am infected. So I give a lecture here. I infect every one of you. That helps it to spread. A virus that is not fatal, but highly infectious. So that is the trend of evolution. It's random mutation, but which one you select?
Well, I have a non-professional question. Can you detect the coronavirus in like other animals, like in lion, tigers, in addition to like camel or bat?
So far, only in mammals and birds, but different kinds of mammals and birds. But of course, we have done limited surveillance studies. I believe that for the mammals and birds that we have not tried to look for coronaviruses, there are probably coronaviruses there, but only mammals and birds so far. Not in reptiles, not in amphibians, not in fish.
Okay, so it's quite interesting because what is the reason why this virus is restricted to those few species? It's not detected in the older animal kingdoms.
Probably because it may exist in a different form. I mean, the virus may exist in a different form because, you know, viruses also evolved just like animals. So some of the genes of coronaviruses have been detected in other viruses as well, not only coronaviruses. But of course, for some of the genes, they are only present in coronaviruses. So like for the hemagglutinin estrates, it's not only present in those coronavirus, it's also present in influenza C. So people believe that there was some sort of exchange for genes between coronaviruses and perhaps influenza C viruses in the past. And those might, this is just an example, and those gene movement might have caused these viruses move from one species to another, one genus of animals to another, families, and so on.
Okay, thank you for your amazing talk. My question is, like coronavirus is widespread from so many taxonomies of animal, and as far as I know, Taiwan also have some bats that carry bad coronavirus. And is there any possibility that we can track back the origin of coronavirus? Maybe they were carried by some animal that migrate from the China or something. Is there any way we can track back the real origin of the coronavirus?
It's also by surveillance study. So the first thing to do is surveillance study, to see what is going on. After we have done the surveillance study, you can set a model. You can set up models to modelize what has happened, what might have happened in the past. In other words, the evolutionary origin. And then you might then carry out further experiments to test whether those modeling are correct or not, or seems to be correct or not.
Because virus don't have, it's just protein. We don't have fossil evidence. So
Sorry, I missed that question.
Because virus just composed with some structured protein and RNA, so we can find some fossil or something from the fossil evidence. Yeah.
Yeah, you are correct. RNAs are difficult to preserve, so you don't find it in fossils basically.
Yeah. Okay. Thank you.
So any more questions? So, yeah, because now it's 4:30, okay, because the seminar is scheduled to 5 o'clock. But however, Dr. Hu speak only for 1 hour, so save our 30 minutes. So if no more questions, then please join me to put your hand together to thank Dr. Hu's presentation today. Thank you very much. If you have any question in the future, pretty sure Dr. Hu will be here, okay? So you can ask him anytime, okay? Thanks again, okay.